Hello friends, welcome to EPG Pathshala. My name is Dr. Priyanka Khurana and I teach Anthropology at the Department of Anthropology, Dr. Hari Singh Gaur University, Sagar, MP. Today we are going to discuss about the module on reproductive isolate and gene pool. This module comes under the paper Human Population Genetics. So let's see that what we are going to learn in this module. In this module, we will learn that what is reproductive isolation. We will learn about the process which leads to the reproductive isolation. We will understand that what are the different types of speciation. By the end of this module, you will understand the concept of gene pool and the factors causing change in the gene pool of a population. Let's begin this module. After Darwin's theory of natural selection, evolution of species became a hot topic. Importance of reproductive isolation and interbreeding started to become an important component while defining the word species. This view is called as biological species concept, and this concept was given by Ernest Mayr. This concept means the description of a species as a group of individuals which are capable of interbreeding but reproductively isolated from one another. The term reproductive isolate is usually applied to a group of organisms that share characteristics feature with the other species but cannot reproduce offspring. It is believed that the two groups evolved to two separate species from a single one due to the development of the sufficient genetic difference between them, which prevents them to mate and reproduce. Let us understand how reproductive isolation or speciation occur in nature. According to the biological species concept, the first step towards speciation is isolation. For example, consider a single species of tiger and imagine that several subpopulation of tiger is distributed over the wide geographical area. Now suppose there is a geographical barrier between the subpopulation which is preventing gene exchange between the population. That means no gene flow is occurring between the two populations. This extremely important form of isolating mechanism is called as geographical isolation. Now, if one tiger population A is separated from another tiger population B by a mountain range or a water body, then tiger of population A will not be able to mate with the tiger from B population. As the time passes, perhaps hundreds or thousands of generations, genetic differences will be accumulated in both the population and eventually they will evolve as a different species. This genetic difference may arise due to the two important reasons. A, if the population size is very small, we can assume that genetic drift may cause allele frequency to change in both the population. And as we know that the drift is random, we would expect the effect to be different in both the populations. Consequently, the two populations will begin to diverge genetically. As long as gene exchange is limited, the population can only become more genetically different over the period of time. Apart from drift, natural selection can also contribute to make these populations genetically dissimilar. If the tiger population are occupying slightly different habitats, then in these populations, certain individuals in population A will be more reproductively fit in their own environment, but they would show less reproductive success in the environment occupied by population number B. So, allele frequency will shift further, resulting in even greater divergence between the two groups. With the cumulative effect of drift and selection acting over many generations, the result will be two populations which are entirely genetically different from each other. The difference is so much that even if when they come back together in a similar geographical area, they could not longer interbreed. So, the process of isolation includes number one, interruption in gene flow between the two populations. Secondly, gradual accumulation of genetic differences between the two populations over the period of time. And thirdly, the divergence give rise to reproductive isolation and therefore to new different species. Let's see what are the different causes of speciation. Speciation occurs when genetic changes take place between previously interbreeding populations of a particular species. These genetic changes prohibit the population to interbreed, leading reproductive isolation and further different species. The whole genetic process involved in the process of isolation lead to the new species and is called reproductive isolating mechanism. In genetics and evolution, shared gene pool is a basis of speciation. By the term gene pool, we means the sum total of all the genes carried by the individual in a population. A group of interbreeding or potentially interbreeding organism that does not exchange genes with other such group is considered to be a species. Each species is reproductively isolated from each other species. It is noteworthy that organisms can be reproductively isolated through easily recognized phenotypic characteristics are not there. So, such organism in taxonomy would be regarded as a single species and in genetics and evolution as a separate species. Similarly, organism may not be able to reproductively isolate but have a different phenotypic characteristic. With fossilized organism, we are limited in the taxonomic definition. Reproductive isolation is the key to the genetic definition of species. Groups of organisms can be reproductively isolated from each other by different mechanisms, even if they inhabit the same territory. From genetic point of view, they are genetically controlled different mechanisms of reproductive isolation. It has been demonstrated experimentally that they can evolve in species whose geographical distribution overlap. Such kind of a speciation is known as sympatric speciation. Whereas, in terms of speciation whose geographical distribution do not overlap are called as allopatric speciation. Ernest Mayr classified the reproductive isolation mechanism into two categories. One, acting before mating or pre-copulatory, and the another, acting after mating or post-copulatory. It is also classified as prezygotic and postzygotic mechanisms. As we have mentioned earlier, there are two types of reproductive isolating mechanism, prezygotic and postzygotic barriers. In these two categories, we have another subcategories. In prezygotic barriers, there are barriers such as pre-mating, habitat isolation, temporal isolation, behavioral isolation, and mechanical isolation. In postzygotic barrier, we have mechanisms such as gametic isolation, postzygotic barriers, hybrid inviability, hybrid sterility, and hybrid breakdown. Prezygotic isolation mechanism prevents the member of different groups from producing hybrid offsprings. Postzygotic isolating mechanism, on the other hand, prevent any hybrid offspring that are produced from passing on their genes to the next generations. In prezygotic isolation, mechanism operates by preventing mating between individuals from different populations of organism, or by preventing the gamete of these individuals from uniting to form zygote. For example, two populations of organisms that inhabit the same area might seek out different habitats within that area. If the habitat preference is strong, the two populations will have little or no contacts with each other. Ecological isolation based on habitat preference can therefore prevent the population from producing hybrid zygotes. Temporal or behavioral factors can also bring about reproductive isolation between populations of organisms. For instance, the organism might become sexually mature at different times, or they might have different courting rituals. If ecological, temporal, and behavioral isolating mechanisms fail to prevent mating between different organisms, then anatomical or chemical incompatibilities in their reproductive organs or gametes prevent them from producing hybrid zygotes. The organism might be unable to copulate successfully, or their gamete might die in the reproductive tissues of their mates. Any of these prezygotic isolation mechanisms could prevent genes from being exchanged between populations occupying the same territory. Postzygotic isolating mechanisms operate after hybrid zygotes have been formed, either by reducing hybrid viability or by impairing hybrid fertility. The zygote from mating between different organisms might not survive, or they might not reach sexual maturity. If they do reach sexual maturity, in some cases, they might not produce functional gametes. Any of these circumstances could prevent populations of organisms that live in the same territory from exchanging genes. Mule is a classical example of post-zygotic barrier. As we have already discussed, there are two types of speciation, sympatric and allopatric. The key event in formation of reproductive isolate is the splitting of a population of organism into one or more subpopulations that become reproductively isolated from each other. The most straightforward way for this event to happen is for the subpopulation to become geographically separated so that they evolve independently. That is, geographical barriers keep the subpopulations apart so that they accumulate their own set of genetic changes over time. Then, if the subpopulations are reunited by the disappearance of geographical barriers, the genetic exchange they have accumulated make them reproductively isolated from each other. For example, one subpopulation may have evolved a preference for a particular food resource, and another subpopulation may have evolved a preference for a different food source. When the two subpopulations are rejoined in the same territory, their distinctive food preferences may limit contact between them to such an extent that interpopulation mating never occurs. Another possibility is that during the time the subpopulation were separated, they may have evolved different physiological processes or mating habits. So, when the subpopulations are reunited, they may not be able to mate with each other, or if they can mate with each other, their hybrids may not be viable or fertile. The process whereby subpopulations evolved reproductive isolation while they are geographically separated is called as allopatric speciation. Allopatric type of speciation leading to reproductive isolate is frequently observed type of speciation in animals. Here, genetic drift acts as a selective force. The effect of genetic drift occurs in all types of population and particularly in population of small size. Here, it is noteworthy that the habitat types of the two populations also play role with respect to the effective role of natural selection. The evidence of allopatric speciation are observed from different types of geographical patterns. When gene flow between populations get reduced due to physical barriers, the geographical populations of the same species can be seen different from each other genetically. For example, the island populations. Another example of species is a ring species. Ring species is a situation in which two populations which do not interbreed are living in the same region and connected by a geographical ring of population that can interbreed. To understand this, imagine a straight line across which species is distributed. It is possible that one form of a species at one end is so genetically different from the another form of a species at other end that they could not interbreed. Now, take this line and bend it like a ring. So you will have a situation where the two extremes meet, but the two forms will not interbreed and the geographical distribution of these species will be in the shape of the ring. This is called as ring species. Around the rest of the ring, the traits of one of these species changes gradually through intermediate population into the traits of the second species. The figure in this particular slide shows the mechanism of allopatric speciation. The steps involved are first of all a population of organism inhabiting a particular territory. In the second step, the geographical barrier arises between the two subpopulations. As a result, these two subpopulations become subdivided and inhabit the different zones. They are no longer in contact with each other. In step 3, the separated subpopulation changes genetically, usually because of the role of the drift over these populations. The drift has an enormous effect because the size of the populations has also been reduced because of their subdivisions. When these populations reunite, the subpopulation cannot exchange genes because they have become so genetically distinctive from each other. Opposite to the allopatric speciation, in sympatric speciation, subpopulations evolved reproductive isolation without being separated geographically. That is to say, the two populations of the similar species lives in the similar geographical area, yet they develop speciation. The subpopulations become ecologically specialized so that they evolve more or less independently, or their members mate assortatively so that there is little or no genetic exchange between these populations. Once the genetic exchange is limited and no longer gene flow occurs between the subpopulation, speciation takes place. As a result, even when these two subpopulations reunite, they are not able to mate or produce viable offsprings. The difference between sympatric and allopatric speciation based reproductive isolate is with respect to how gene flow occurred. In one, the gene flow is prevented due to geographical barrier, whereas in another, the gene flow is prevented due to ecological specialization without being geography acting as a barrier. The first one is known as allopatric speciation and the second one is called as sympatric speciation. The figure given in this slide shows the mechanism of sympatric speciation. As we can see in this particular slide that a population of organism inhabits a territory. Initially, there is no speciation. Later on, within the same geographical area, subpopulations are formed. These are formed due to ecologically different adaptations. These adaptations to different ecological niches within the given geographical area leads to make these species genetically different. Once the genetic difference has arisen between these subpopulations, they become reproductive isolate, and mating between these two populations initially belonging to the same species is not possible, and each subpopulation becomes a distinct species. Example of sympatric speciation is Myzomela honey eater from Pacific. Another kind of a speciation is parapatric and parapatric speciation. Parapatric speciation leads to development of isolate when the new population is smaller than the original population. In parapatric speciation, small population is isolated from the main population, and this small population makes full-blown populations. In this type, both geographical isolation and genetic drift plays a crucial role. Contrary to parapatric speciation, parapatric speciation happens when there is no physical barrier between the subpopulation. Rather, there is a difference in the common environment of the species. That is, the population are not separated by any geographical barrier, but by an extreme change in the habitat. As a result, population develops distinct characteristics feature and become reproductively isolates. One of the famous examples of parapatric speciation is from the area of mining activities. In these areas, lead and zinc-like metals are present in the soil. Though this element prevents most plants to grow, buffalo grass could grow and adopt the soil type. This type of grass is found in many parts of the world, particularly known from the mining areas. One of the fundamental questions in evolutionary biology is what type of genetic changes bring about reproductive isolation between populations of a species. This question is difficult to address because if the population are reproductively isolated, they either cannot be crossed, or if they can be crossed, their hybrids are sterile or unviable. In experiments on Drosophila, that is fruit fly, it has been found that the difference is more quantitative than qualitative. Dobzhansky and Muller has given an explanation of the genetic changes. According to them, the viability or the sterility of hybrid is due to incompatibilities between genes that evolved separately in different populations. For example, if a population with the genotype AA, BB becomes subdivided by geographical barriers, one of its subpopulation might fix a new allele on the locus A, and another population might fix a new allele on the locus B. If these subpopulations rejoin after the disappearance of the geographical barrier that separated them, they might cross to produce hybrids. Such crosses would bring new alleles together in the same genotype for the first time. Because these alleles evolved in a separate population, they might interact unfavorably with each other and causes death or sterility. Thus, the independent evolution of genes in separated population might be the reason of unviability or sterility of the offsprings. Different breeding experiments on the species of Drosophila have led to the another conclusions. Chromosomally differentiated population can also give rise to the post-copulatory isolation due to chromosomal translocation and inversion. For example, a reduced fertility with chromosomal aberration arises due to incomplete meiosis when a reciprocal translocation is fixed in a population. Such example of chromosomal changes leading to reduced fertility can be drawn from the two twin species of Drosophila from the Indo-Pacific region. Presence of microorganism in the cytoplasm can also act as another factor for postzygotic isolation. Again, taking example from the Drosophila species, it has been observed that the female Drosophila of a particular species hybrids are fertile while males are sterile. The males contain a mycoplasm in the cytoplasm that can reduce them to sterile. Now let's move towards the forces of gene pool change. These forces are same which brings about the evolution. These are natural selection, random mating, mutation, gene flow, genetic drift, such as bottleneck effect and founder's effect. To begin with, let's discuss mutations first of all. Mutation, as we all know, is the random change in the actual genetic code, including changes in single DNA base. It can be insertion or deletion of DNA sequence or any other rearrangement of DNA sequence. The next one in the line is natural selection. Many processes changes gene frequency independently of their increasing or decreasing adaptive power. Natural selection is one of the processes that acts to change the allele frequency of a population. It is a process that promotes adaptation and keeps in check the disorganizing effects of other processes. Natural selection is thus most critically evolutionary mechanism that affect the gene pool of a population. Environmental changes promotes genetic changes, and once these genetic changes have been established in a population, no more mating occurs between the population and speciation is established. Another force which affects the gene pool is genetic drift. Population of organism consists of limited number of individuals, though it may be extremely large in some cases. The small size of population causes chance alteration in allele frequencies. This chance changes in the allele frequency that results from sampling of gametes from generation to generation in a finite population is known as genetic drift. Unlike selection, whether an allele is good or a bad, its frequency changes abruptly from one generation to another. Genetic drift has same expected effect on all the loci in the genome. Genetic drift is classified into two categories, one being bottleneck and another being founder's effect. Drift can occur because of chance departure of Mendelian expectations in a few families or because a newly formed isolated group is not representative of the original population. This type is known as founder's effect, where a group of the founding members get separated from the main population and establish a new population. Another special case is a bottleneck effect, where a community sample of an allele is changed due to natural catastrophe, disease, or other factors. In a large population, on an average, only a small chance change will occur in the allele frequency as a result of a genetic drift. However, in a small population, these effects are much more conspicuous. The another one in the line is a gene flow. Unlike drift, natural selection, and mutation, which causes population to differentiate genetically, gene flow causes population to homogenize. So, if the speciation has not been established so far, gene flow do not let the speciation to occur by introducing genes of one population into the another population and making them more homogeneous. Now let's understand the concept of gene pool. Gene pool is the total complement of genes shared by the reproductive members of a population. For example, imagine a population of a frog in which most are green in color, but some are blue and red. So if you consider all the color together in a population, we call it as a color pool. Now apply this idea to genes. So gene pool is a sum total of all the genes and their alleles that exist in a population. In every generation, the genes or allele in a gene pool are mixed by recombination and then reunited with other of their counterparts through mating. What emerges in the next generation is a direct product of a genes going into the pool, which in turn is a product of who is mating with whom. Factors that determine mate choice are geographical, ecological, and social. Such breeding isolate populations that are clearly isolated geographically or socially from one another breeding group are fairly easily defined and are favorite target of microevolutionary studies. Geography plays a dominant role in producing these isolates by strictly determining the range of available mates. But even within these limits, cultural rules can apply a deciding role by prescribing who is the most appropriate among those who is potentially available. Once specific human population have been identified, the next step is to ascertain what evolutionary force, if any, are operating upon them. To determine whether evolution is taking place at the given locus, population geneticists measure allele frequency of a specific trait and compare these observed frequency with the set predicted by the mathematical model. One such model is Hardy-Weinberg equilibrium equation. There are many factors which can have pronounced effect on the gene pool. So students, let us summarize this module. In this module, we have learned the concept of species. A species is a group of organisms that can interbreed to produce fertile offspring. Members of one species are reproductively isolated from members of the other species. That is, they cannot mate within them to produce a fertile offspring. Reproductively isolated means pertaining to a group of organisms that, mainly because of the genetic differences, are prevented from mating and producing offspring with the members of other groups. There are many processes of reproductive isolation, and the step involves basically are: number 1, interruption in the gene flow between the two populations. Secondly, gradual accumulation of genetic differences between the two populations. And finally, this divergence gives rise to reproductive isolation. Factors causing reproductive isolation. There are many factors which can prevent interbreeding among populations. They can be pre- and post-zygotic factors. These pre- and post-isolating mechanisms may include geographical barrier, ecological barrier, behavioral isolating mechanism, anatomical and chemical incompatibilities with each other. There are different types of species and the process of speciation. The one we discussed in this module is sympatric species. This is the process whereby subpopulations evolved reproductively isolating with each other while they are geographically coexisting. Allopatric species, on the other hand, is a process whereby subpopulations evolved reproductive isolation while they are geographically separated. That means in sympatric species, geographically they coexist. In allopatric, geographically they are disparate. Ring species. A ring species is a situation in which two populations which do not interbreed are living in the same region and connected by a geographical ring of population that can interbreed. Parapatric species is a species where small population is isolated from the main population and makes a new population. Parapatric species, on the other hand, is a kind of a species where population is not separated geographically but by extreme changes in the habitat. There are many causes of speciation which result into the change in the gene pool. Gene pool is a total complement of the genes shared by the reproductive members of a population, and factors like natural selection, random mating, mutation, gene flow, and drift plays an active role in changing the gene pool of a population and thus causing and preventing speciation. Thank you.